Good morning, Namaskar. Welcome to Hortalk-53. Today's speaker is Dr. Popy Bora. She is from Assam Agriculture University, Jorhat, Assam. She will speak on microbes and disease management in horticultural crops. On behalf of Progressive Horticulture, I, Shiv Pratap, welcome the speaker Dr. Popy Bora and you all to this edition of Hot Talk. Now I request Dr. Popy Bora to start the talk. Please, ma'am.
Thank you very much. Yeah, thank you very much, and very warm good morning to all the viewers. This is Popy Bora from Assam Agriculture University, and as you know, that Assam Agriculture University is one of the premier educational and research institute in entire Northeast India, and it is a proud recipient of Sardar Patel Outstanding Award in 2014. So I have started my career in Assam Agriculture University as subject matter specialist in 2009. Since then, I have been serving in teaching and research, and presently I am engaged in ASAP on fruits and also associated with teaching in plantology. The topic for today's discussion is disease management, microbes and disease management in horticultural crops. Of course, it is a very fascinating area. Microbial world itself is a very fascinating world. And how I generated the interest in microbes. There is a small story. Without wasting much of your time, let me share with you one particular incident of my childhood, which led me to enter this particular world of this particular stream. During that time, probably I was in third or fourth standard. Exactly, I forgot. Uh, I entered the kitchen in the evening and I could see a very strange insect. It was somewhat like your spider, but maybe it was not spider. Color was different and its body structure was different. And during that time, I had some phobia on spider phobia, you can say. So I screamed and I came out. And my grandma, she then came in and she asked me what happened. Then I, then I told her what I have seen exactly. Then she took me to the kitchen and she shown me that was a spider preying on a cockroach. So she told me that some spiders, they are our friendly spiders, and which clean our homes from, notorious pests like cockroaches. That day, I got two lessons in my life. I could overcome the fear of your spider phobia. And she taught me one very important lesson that is nature has solution for every problem. So eventually when I joined my BSc Agri program and MSc Agri program, I came across the terminologies like your biological control, natural bioagents, predator parasitage. So then this reminded me of my grandmother. Then I wish I could tell her what she taught me was a fascinating branch of science. So this particular incident generated the interest in microbes, and I got an opportunity to pursue my MSc and PhD in plant pathology with specialization in biological control. So today I'm thankful to my grandmother, and today, most importantly, I'm thankful to the society and progressive horticulture for giving me an opportunity to talk on this particular topic, microbes and disease management in horticultural crops. So now, without wasting much of your time, let us get back to the business. When we talk about plant diseases, we know what impact of plant disease is there in human civilization. Two major incidents, two major tragedies, you can say. The Irish famine and the Bengal famine has taught us very important lesson about the impact of plant diseases in socioeconomic nation. So plant diseases are the major biotic factors. You can say limiting factors for reduced productivity of all the crops, including horticulture and field crops. When you talk about disease management, there are a lot of, other disease management practices are there, like chemical, biological, cultural. But as a quick fix solution, farmers always go for chemical management. And during the post-grain revolution era, so disease management, it was basically the chemo intensive. based. The situation has been changing in recent time due to your enhanced our knowledge on ill effect of chemicals, enhance our sensitivity towards environmental degradation, our health deterioration due to chemical input. So disease management and pest management as well, it is changing towards some biointensive approaches. So in this regard, microbes are a major key player, we can say, for disease and pest management also. This is relevance of microbes or relevance of biointensive pest management is very really, important in all the crops, especially in horticultural crops, because everyone wants to have a residue-free tea in the morning. Our fruits and vegetables, basically, we want to have some residue-free or, if possible, organic ones. So, if you see the world scenario, the demand for organic horticulture, organically grown crops, food commodities has been increasing day by day. And in this regard, I would like to show you some of the statistics. Like India, it is the largest producer of organic crops, organic products, with around 3.67 million hectare. And MP is the maximum uh, area. MP is having the maximum area. And production wise also, India produces around 2.75 million metric tons, and it exports 6.389 lakh metric ton. And this is from, of course, EPIDA website. And out of this export, majority is of basmati rice and some are dry fruit, tea, and spices. And if we consider organic production of these commodities, and if we consider production of some residue-free horticultural food commodities, definitely the mass focus will be on the microbial-based production system, you can say. So in this regard, microbes can play a better role in plant protection. So if you see the biopesticide use trend in global scenario, you can see that around 1400 biopesticides are registered all over so far, and out of all this, 101 microbial biocontrol agents are registered in EU alone. And BT here is a key player in biopesticide market, and North America, it represents around 44% of the biopesticide market. And if you see the Indian scenario, we are also not lagging behind. We are having right now 970 biopesticide registered, and of which 355 are trichoderma based, and rest of the others are like other microbes, Pseudomonas, Bacillus, like that, and some entomopathogens. So these microbes have long been used for disease management, and relevance is more in the current scenario for organic production or sustainable production system. So these are some of the microbes basically being used for disease management. Some bacterial and some fungal. Out of bacterial, Bacillus, Pseudomonas, and out of fungal, Trichoderma, Aspergillus, Penicillium. These are commonly used, and all these biocontrol agents, they are mostly successful against some soil-borne pathogens like root diseases, damping of.
Excuse me, ma'am. Ma'am,
Yeah, sure.
put your slide full mode, ma'am. Full.
Okay.
Thank you, ma'am.
Okay. These are some of the biocontrol agents being used worldwide for management of some major diseases, and mostly the soil-borne diseases and some foliar leaf spot diseases. And it is worth mention in this field that these microbes have long been used, and there is some various, success stories of disease management with these microbes in horticultural crops, like Agrobacterium tumefaciens. It causes crown gall disease, and which was a major problem in apple pear like crops, and it was successfully managed by Agrobacterium radiobacter, a non-pathogenic strain, and which produces a particular bacteriocin, and With that, it was successfully managed world over through this agrobacterium radiovector strain K84. And another success story was chestnut blight. It was an American tragedy. Chestnut trees were severely infected by a fungus, Endothea parasitica. The name is now changed, and that was in early 1900s. Around 1903, it was reported, the fungus was isolated and reported. And later on, it was managed through a hypervirulent strain of the particular fungus. And it was an uh, hypervirulent strain, it was an infected strain by a double-stranded RNA virus. So these are some important case studies where horticultural crop disease management was successful through biocontrol agents. Now, question is that where from these microbes come? So, let me give you one statement here. Someone said, or I read somewhere, a plant standing in the field is not an individual; it's a community. And I believe this statement is scientifically very correct because a plant in and out, it carries a diverse group of microbes in the rhizosphere and the endosphere. So some are beneficial and some are of course harmful. And during this interaction process between plant and microbe interaction, the beneficial microbes are being explored for plant disease management. Okay, uh, here comes another one question. Why uh, microbes they move towards plants? And as we all know that in the rhizosphere, due to rhizodeposits, the secretion by the root exudates, they attract the beneficial microbes. And other than these non-natural colonizers, and among these, there may be some harmful, there may be some are beneficial. But a very interesting phenomenon some scientists have, discovered like some plants during your pathogen stress condition, they can recruit beneficial microbes of their own soils to manage the diseases by secretion of some particular chemicals. Here you can see one table I have shown some plants they secretes more flavonoids, which attracts beneficial microbes like rhizobia, and some secretes jasmonic acid signaling specific exudates, some they secrete stregolectin, and some secrete malic acid, succinate acid, fumarate acid, and which invites more of the beneficial microbes of their source of a particular community. And the result you can see mostly plant growth promotion, nitrogen fixation, then induce host resistance and induce nutrient uptake and separation of different pathogens. So how they interact, what is the crosstalk between microbes and the plant, it is still yet to be uncovered. And coming to the different modus operandi of biological control agents, so we all know there are different mode of action, and these mode of actions are particularly used for screening of microbial biocontrol agents like hyperparasitism. These are some direct mode of action like hyperparasitism, antimicrobial metabolites like antibiotics, some volatiles, some toxins. And hyperparasitism, we know that when a particular pathogen it parasitizes another organism, that is hyperparasitism. And trichoderma is the best known hyperparasite against many soil-borne pathogens like rhizoctonia, pythium, and fusarium, of course. And another mode of action is competition. In competition, what happens? Microbes, while interacting in the soil, they always face deep competition for nutrients as well as for site of colonization. And as a rule of thumb, survival of the fittest, always those microbes which can efficiently utilize the nutrients there, the winner, and they suppress the pathogens. And the best known example is trichoderma. When we go for dual culture assay, we can see that trichoderma is always overgrowing, and it this way also, and through other mode of action also, this outcompetes the pathogenic microbes. Same phenomenon happens in the soil also. And another very important feature for competition is for competition for iron. You know that iron, although it is one of the most abundant molecule, but it is not in not soluble and not in available form. And some of the microbes, some of the pseudomonas fluorescens and some bacillus and some fungus also, they secrete a particular uh, group, I mean siderophore, which binds with a ferric iron, and this complex is again retained back to the bacteria or fungus which produce it, and this way they deprive the pathogenic microbe of iron. And this is another mode of action. So hyperparasitism and antimicrobial metabolite production, these are always selected for screening during our screening procedure of a biocontrol agent. But thing is that while screening the biocontrol agents, the competition part is always overlooked. Other than your siderophore, siderophore is in production is an important criteria for selection of, I mean, microbial bioagents. But some beneficial microbes, they are active nutrient explorer, you can say. Uh, Other than your obligate biotropes, which are dependent on the host nutrition, but other microbes, they mainly depend on the soil nutrients. Those which can efficiently use the soil nutrients, they should be selected for uh, biocontrol of may be selected. This can also be a criteria. If a particular microbe it is very much aggressive in utilizing nutrients, that way and doesn't allow other pathogenic microbes to grow, then also it is definitely very much useful for us. And that can, if the particular microbe is having ability to produce different antimicrobial microbe, I mean agents, or if it can hyperparasite other uh, pathogenic microflora, then definitely it will be uh, sone pesohal, I can say. So these are mode of action and based on which the microbes are screened. But other than this mode of action, pathogen, I mean, beneficial microbes, they also induce resistance in the plants. This is a indirect mode of action of biocontrol agents. Here I am showing you one pictorial representation, the plant rhizospheric microbe crosstalk and disease separation. In plant, usually resistance is a normal phenomenon and disease is a rare phenomenon, you can see. Disease is abnormality. Resistance is normal for plants. So plants, when silenced by pathogens or microbes, pathogens and beneficial microbes, both are foreign bodies for a plant, but the plant perceive both the things in a different way. In pathogenic attack, what happens uh, when the pathogen PAMP, you can see, pathogen associated molecular patterns, these are recognized by some cell surface receptor present in the plasma membrane. These are called pattern recognition receptors. This is the first line of defense, and then they activate the defense mechanism. Plant has a very sophisticated defense mechanism, and but some microbes, some aggressive pathogens, they can overcome this first line of barrier, and they secrete some effector molecules, and that is again in resistant plant. There are R-coded genes like NBLR. They again recognize that effector proteins, and they again activate the second line of defense, and like your SAR. Okay, but in case of your microbial beneficial microbe, when they interact, then microbe-associated molecular patterns, the mumps, they also induce different response in the plants. Of course, it is somewhat different, like your pathogen induced, and earlier it was thought that microbe, the beneficial microbe, they induce systemic resistance, and desmonic acid or ethylene is the signaling molecule. But the recent study says that even the salicylic acid-based pathways also being followed by plant beneficial microbes. In this case, when the beneficial microbe, the mump is recognized by the PRR, then of course there will be oxidative burst and cellular thickening and accumulation of PR proteins, and before that, salicylic acid production, and that will be the signaling molecule for activation of defense cascade. So this is how the beneficial microbes uh, defends the plants from pathogenic attack. So let me tell you one more important, interesting feature I could get somewhere in some recent literature, that some, microbes, especially those which are recruited by pathogen, they induce the host resistance, and that has been transformed, or that has been passed on to their progenies. And this is called transgenerational resistance. And how it happens, still it is a matter of research. So this was all about, I mean, rhizospheric microbes. The scientists have recently explored another group of microbes, which is called endophytes. And endophytes are nothing but, this is not new, in fact. They are one sort of your rhizospheric microbes only. In the some part of their life, they live inside the plant tissues, or a complete part of their life, they live inside the host tissues. And this definition says that the microorganisms that live within the plant tissues for all or part of their life cycle and cause no apparent infections or symptoms, it is called endophyte. But recently, another group of scientists declared that microorganisms that live in the plant tissues, the whole or part of their life cycle, but irrespective of whether they cause disease or not, they are endophytes. But that is not a topic of discussion for us. We'll simply uh, concentrate more on the beneficial ones. So these endophytes also, this is symbiotic association gives benefits to the plant as well as to the microbes, and these are benefits the plant receives, like disease management and reduction of host resistance, and production of growth regulators, biological nitrogen fixation, phosphorus solubilization, drought tolerance. In short, you can say that it gives protection from biotic and abiotic stresses, including your disease and some pest management also. And what still there remains a question always comes to my mind. Endophytes also is a foreign body. While endophytes enter the host system, why plants do not defend themselves? Actually, this phenomenon is still yet to be uncovered. Why host plant, they react differently for endophyte and harmful pathogens. How they differentiate, it is still to be uncovered. But still, some literature says that the mump, when it induces your reactive oxygen species, the reactive this environment, some endophytes, some bacteria, they can survive in the oxidative burst condition, because they can produce some of the enzymes like dimutase and hydrogen peroxide reductase. This way, they can neutralize the effect of oxidative burst. And some endophytes, they do not secrete effector molecules to further activate the second line of defense. This way, they can survive inside the host system without uh, causing harm or without being harmed by the host. And still, a lot of things are yet to be elucidated. So these are some of the endophytic bacteria isolated from some horticultural crops like radish, canola, citrus, black pepper, onion, tomato, and chili, banana. Here you can see that these endophytic microbes, like mostly of Pseudomonas and Bacillus, and some actinobacteria and trichoderma also, they have been identified, they have been isolated, and their roles have been established in the host plant, like induction of systemic resistance or suppression of some pathogens. So coming to disease management of horticultural crops with biocontrol agents, as I had already shared that some of the diseases have successfully been managed through biocontrol agents. Here is the table of some bioagents which were found successful in vitro and in vivo condition against some pathogens, and mostly of these pathogens are fungal wilt, bacterial wilt, then fruit rot, root rot, and damping of anthracnose, like this. And mostly are some soil-borne wilting, rotting, then fruit rot type of diseases have been successfully managed. And these are for vegetable crops. And for spices and plantation and aromatic crops also, it is very much important for export point of view. Where you can see in the ginger rhizome rot is a very severe disease, and it can be successfully managed through Trichoderma viridie and Pseudomonas fluorescens based bioformulation. Then again for cardamom clump rot can be managed by Trichoderma. Phytophthora rot can be managed by Trichoderma, and black pepper also. Similar way, fennel damping of can be managed by some biocontrol agents, and vanilla and tea, the most important export oriented crop. Uh, Tea, there are a lot of pathogens, but the pathogens, the diseases of foliar diseases are very much important because the tea leaves, the young buds are the main, part for harvesting. And grey blight disease, it is one of the very severe disease of tea, and it is successfully being managed in our university through application of Trichoderma viridis, Bacillus, and Pseudomonas fluorescens based bioformulations. So these are some successful examples of biocontrol of plant diseases of horticultural crops. Here, I would like to uh, share our experience in microbial bioformulations developed at AU Jorhat. But before that, uh, let me tell you there is a proverb like, united we stand, divided we fall. This is, of course, true for everyone, but during this COVID pandemic, this is not relevant to us, but it is still relevant to the microbial community. Plants, they harbor a lot of different groups of microbes, different antagonists in the rhizosphere, in the endosphere. They have different mode of action. If the compatible microbes with different groups and different mode of actions can be made a consortia formulation, definitely their efficacy will be much more. So in that way, we lot of different bioformulations, consortia formulations have been developed. And in our university also, we have been working on some specific biocontrol agents, and we are trying to develop some of the consortia formulation for management of different diseases as well as insect pest. So these are some of the bioformulations developed at our university. This is Trichoderma viridie-based biovir, then Biomonas, it is fluorescent, Pseudomonas fluorescens based, then Biometa, Biosona, and Biolium. These are entomopathogens based, and Biofilm PF2, this one, it carries revival cells of aggressive cells of Pseudomonas fluorescens and Trichoderma viridie, and this last your two formulations, Biozin PTB and Biogreen. Here you can see more than one microbial agents. In Biogreen, even you can see Pseudomonas fluorescens, Trichoderma viridie, along with that some entomopathogens are also there. And these are found very much successful under field conditions. So in our university, this research on disease management through biocontrol agents, it started long back in 1972, but a commercial formulation, it was the Biofor PF. The whole story began with Biofor PF1. Initially, it was Pseudomonas fluorescens based, and later on, it was updated, you can say, with Trichoderma viridie. And it was a huge success for management of bacterial wilt disease in Solanacea crops. While giving demonstration and trials and trainings on these biocontrol agents, bioformulations, we frequently encountered some questions from farmers. Farmers used to always ask, because in field it is not that there will be only diseases, there is infestation of insect pests also. So in that case, what we can do? Is there any single solution? Is there any single formulations which will address both the insect pest and disease aspects? With that question, we could drive our research in a different angle, and we try to study the compatibility between some microbial, bioagents for disease separation and some entomopathogen, and these are results of those particular studies. So presently, we are using these microbes in our research and development of microbial biopesticides. And of this product, Assam Agriculture Industry Jorhat has signed MOU with 3 private sectors, and 2 of our technologies, BioFur and BioGreen, has already been transferred to private sectors for large-scale production. This is simply what I said. Like the plants face multiple challenges in the field: insect pests, abiotic sprays, then other things. They are major limiting factor for any crop productivity. And addressing the diseases and pests with a single formulation needs a consortium approach, and this is what we are trying to do. So our BioGreen technology, it has been a very successful one, especially in tea, and it addressed many soil-borne diseases and some foliar diseases like grape blight of tea. And at the same time, it is found very successful against pests of tea like red spider mite, looper caterpillar, tea aphids, and tea mosquito bugs. And this way, it has been found successful against some citrus pathogens also, citrus insect pests also. So this one is one of the success story of our agriculture university bioproduct. So with our bioproducts, we just try to evaluate the quality aspects of our bioproduct and some other commercial formulations. Here you can see that our own bioformulations, they are far better than some commercial formulation in sense of your population dynamics of Trichoderma viridie. These microbial biogens, as we all know, that they also have some downgrowth promoting properties. They can enhance seed germination, and in this study, we could found that this all these trichoderma-based formulations, they could enhance germination, but our BioVir and BioFur PF2, they were more effective in enhancing germination of just some tomato seedlings, and the vigor index also, you can see it increased the vigor index as well. So another study which are bioformulations, we could found that it was against heterus canker disease. We found that our bioformulations were applied in soil and soil plus foliar application. There we could find that this BioGreen, which consists 5 different microbial bioagents, it was most successful in reducing the disease incidence, and accordingly fruit yield was increased, and it was found that around in BioGreen, when applied as soil and foliar application, it increased the fruit yield by 41.90% over control. So, as I have shared, that our formulations have been found quite effective against tea diseases, and here I would like to show you one other research study where we evaluated some of our bioformulations, BioGreen and BioMeta, against grape blight disease and some insect pests. You can ask me why BioMeta, because BioMeta, it is an entomopathogen. But during our initial study, our preliminary in vitro screening, we could found that BioMeta, although metarism and anisoply, it is considered as an entomopathogen, but it also showed very encouraging result in suppressing the pathogens under lab condition. That means each antimicrobial property, as an antagonistic microbe, this property is still overlooked and it needs some urgent attention. So in this study, we could found that the combination of these 2 bioformulation, BioGreen and BioMeta, they could reduce the disease incidence from around 60% to around 10%, and other bioformulations also showed promising result, and botanical formulation also could show some significant results in reducing the disease incidence. Of course, the efficacy of microbial bioformulation in biograin and biograin plus biometa, it was much higher than botanicals. So we studied the defense response, the microbial biogens and botanical initiates in the plant system. Here we could found that all the botanicals and bioformulations, they enhance some defense related enzymes, activity of some defense related enzymes like PAL, PPO, PO, and beta-1 glucanase. And here in this pictorial diagram, you can see that, and the concentration, the activity of the PAL enzyme was much higher in all the treatments in 0, 24, 48 hours. In 48 hours, it was much higher. It increased and gradually it started declining. But in case of control, it remained almost static. And similarly, in case of PO, PPO, and beta-1 glucanase, they enhance activity of all these enzymes gradually, and it reached maximum in 72 hours. Then it started declining. This is simply to show how microbial bioagents they can enhance productivity by reducing diseases and directly through antagonism and through activating the plant defense cascade. So this is the field view of effect of different treatments in the tea plant. Here you can see the very green luster in the tea leaves, and this is in response to our bioformulation. This is in response to botanicals, and in control situation is completely a different one. So this is all about our university. And coming to another very interesting success story of biocontrol agents in management of diseases like Fusarium wilt TR4. We know that presently TR4 of Fusarium wilt, it has become a major threat. It has shaken the banana industry worldwide. And India, it was reported in 2015. In right now, it is found in UP, MP, Gujarat, and UP, MP, and Gujarat, I think. And this particular disease was successfully managed by one technology developed at ICR-CSSRI and ICR-CSH, and technology was called ICR Fusicount. And this is nothing but a biocontrol agent that is a new strain of Trichoderma rhizae, and this particular technology could save the banana from tier 4 in different part of the country where it was found very aggressive, like in Uttar Pradesh and Bihar. So here you can see the effect of Fusicon technology. Here is the infected plant, and this is the effect of Fusicon technology. Right, from disease management and the yield enhancement, it was found quite encouraging, and we are quite hopeful this could be the permanent solution for tier 4 management in coming days. And these were some of the disease management pictures for field crops and horticultural crops. But other than this, the crops also face the challenge of some post-harvest pathogens. And post-harvest pathogens like Herbaphraitis, Penicillium, Botrytis cultitrichum, and Penicillium, they are one of the major reasons of post-harvest losses. And these could be successfully managed through some biocontrol agents. Here I have shown you some of the list. Basically, the bacillus and some yeast are found very successful against the post-harvest diseases. But the study of disease, post-harvest disease management through microbial-based technology, it is still in very nascent stage. In another study, we could found that when we apply the biocontrol agents in the for post-harvest disease management, what happens? The post-harvest pathogens, they increase the acidity of they produce a lot of acid and increase the acidity, and for which, as a result of which, the microbes, the beneficial microbes, they cannot withstand, or their efficacy is reduced to a great extent. And to encounter this problem and to combat this problem, some salt additives were tried and were found very effective in enhancing the activity or efficacy of the biocontrol agents for management of this post-harvest pathogens. So here you can see the salt additives like sodium carbonate, sodium bicarbonate, ammonium molybdate, and calcium chloride and potassium metabisulfite, and these were found very effective in enhancing the activity of plant beneficial, antagonistic microbes. But the doses and the method of application are yet to be standardized. So this is in short about the disease management through biocontrol agents. There are a lot of things scientists have already done, a lot of things are being done, and a lot of things are yet to be done. A lot of, mechanisms are yet to be uncovered. So, coming to the end of the talk, let me highlight some of the issues we need to address in in near future or in immediate time. So, first one is better screening bioassay methodology to decode the interplay between overlapping mode of action. As I told you that the competition is almost overlooked, and competition as a criteria for selection of beneficial microbes, it is mostly overlooked. And a three-partner model system is required, mostly required to understand the interaction between pathogen, the putative biocontrol agent, that means the host, and under a set of we have to identify the set of genes responsible for biocontrol mechanisms so that we can develop some high throughput screening process for selection of the biocontrol agents in near future. Because if you see the scenario, the pesticide, biocontrol agents are mostly of trichoderma, pseudomonas, bacillus, and there is no such novel, biocontrol agents have been identified. For that reason, our screening methodologies should be somewhat changed, I feel. Then second thing is that application of multi-omics for better understanding of plant microbe interaction. As I told you that few things are yet to be decoded. How endophytes survives in the host system, and how plants, some microbes, they recruit their desired microbes in the pathogen challenge conditions, and how they differentiate those pathogens from, beneficial microbes from others. These things are yet to be elucidated. Further, multiomics is the best tool for understanding this plant microbe interaction, and ultimately we can get some clue for identification of some novel strains. Then, role of microbial antagonists in post-harvest disease management. Although a lot of research have been done in the last decade, but still there is no packaged technology for management of post-harvest diseases through microbial bioagents, and this needs an urgent attention. And also, as I shared the slide, that how salt can enhance the activity of bioagents, and the dose, the method of application, and that has to be standardized. Again, your rhizosphere hybridization. It is a frontier area of research because you can see, forget about the uh, suppressive soil, but in the suppressive soil also, there are some particular plants, there are some particular niches where there are a lot of, microbial activities, beneficial microbial activities, and rhizosphere hybridization of those, microbes, those rhizospheric soil, with some very conducive one, we can think of some rhizospheric engineering, and for that also we need some omic tools. Then development of biofungicide and nano-based technology-aided microbial bioformulation. There are a lot of bioformulations have been developed, registered so far, and nano-based technology are yet to be, yet to be an add-on component. And here I can give you an example of your citrus greening disease. In citrus greening disease, we know that the pathogen, being a phloem inhibiting bacteria, it blocks the vessels, nutrient uptake, nutrient mobilization. It is almost blocked. And if we can introduce some nano particles, nanocomposites. That way, the plant can take up those nanonutrient particles, and at the same time, nano silver, nano copper like things. No, if we can introduce, then they can be, they can work as bioactive, I mean, agent against pathogenic, against the bacteria. And lastly, development of consortia to address biotic and abiotic stress in a single go. As I shared, like plant face multiple challenges, right from diseases, right from your insect base, and some abiotic stresses like your drought, alkalinity, acidity, and heavy metal. And some endophytic microbes, they have they carry the genes for bioremediation of some pesticide polluted soils, for some heavy metal polluted soils. That has to be identified, and if possible, if they are compatible with the biocontrol agents, we can think of development of some consortia formulation so that we can address maximum issues with a single go. So there are a lot of scopes, a lot of things to be done in the near future. So with this, let me conclude my presentation, and thank you all for your patience hearing. And I sincerely hope that in the coming days we'll come out with a lot of novel strains. We'll come out with some understanding about the host microbial interaction process, the signaling molecule, and all. And there will be a lot of practical utility of microbes in plant disease management for organic horticultural crop production as well. So with this, let me conclude, and thank you very much.
Thank you, ma'am. Thank you a lot. Ma'am, we have a question.
Yes. What is your opinion? Endophytes are better colonizers than rhizosphere microbes? I would say yes, because endophytes colonizing in the outer environment, I mean, in the endospheric region, the rhizospheric region, and colonizing in the internal tissues are somewhat different. In internal tissues, plant tissues, as I told you, that plant activates the defense mechanism. But endophytes, they overcome those defense barriers. That way, they are active, more active colonizers. And another, my observation is that some wild plants, they have luxuriant growth during your disease epidemic also. But the reason behind, I feel, other than the genetic resistance, they probably carry some microbes within themselves because microbes are co-evolved with the plants. So in that sense, I think they are better colonizers.
One more question, ma'am.
Hmm. Yes, is there any organism for control of rhizoctonia and fusarium, rotten turmeric, and ginger? Of course, sir. All fusariums are successfully managed, and rhizoctonia also through trichoderma. Even pseudomonas fluorescens also works better. So any trichoderma formulation or trichoderma plus pseudomonas formulation gives better yield. I think, sorry, I got an answer. Whether functionality of beneficial microbes and antagonists run complementary to one another or not necessary. Whether functionality of beneficial microbes and antagonists. I'm not clear about the question, sir.
Others question we will send you by mail, ma'am.
Okay, thank you. Most welcome.
Thank you for wonderful talk, ma'am. Audience, please send your query and question to progressivehorticulture@gmail.com. Response of audience was tremendous with very encouraging remark. We are highly grateful, madam, to you for sparing time and delivering such an important talk. Thank you, audience, for your active participation and support. Thank you all.
Thank you very much for giving me the opportunity to come to this platform, and thank you, audience. And at the last, I would like to say very happy New Year in advance to you all. Thank you.
Happy New Year, ma'am. Thank you. Thank you a lot.